Good afternoon, everybody. Hope you're not missing school too much. I'm sure you're missing the crack anyway. I know I am. I'm missing hearing what you've been up to. But I suppose we have to get on and do some work. So I'd set you some work on immunity because, well, the first topic we do when we go back, usually in September, is the kidney. And then the second one is immunity. But because obviously with the coronavirus in the news, I thought it'd be good just to recap on some of the immunity you'd done at GCSE and look ahead to the kind of things you're going to be talking about come sort of October, November time. So, obviously I hope everybody's doing well. I'm doing well. The only thing I could do with is a decent haircut, but I think we're all the same. It's been good to hear, that you've been able to spend time taking up your new hobbies and getting back to other ones, maybe helping out in farms and so on. Some of you have been baking, so that's very encouraging. Looking forward to some of you bringing in some of your cupcakes and things like that whenever we have coffee time next year, and hopefully somebody will remember to bring milk and coffee and things like that, but that's something to look forward to. So to get back to the immunity stuff, probably the best thing to do is bring up the questions and your answers to the antibody test assignment. So I'd given you a news article to read that had been just published by the BBC that day and telling about a new antibody test, because testing for the coronavirus has been in the news a lot. They're talking about testing and tracking and tracing and so on. So before I give you answers here, could I just say I am not an expert in this. There is an awful lot of information out there, and I know you all researched well and you were getting information from different sources. Just be careful, or just be aware of the fact that what's on the internet could have been written by anybody, and sometimes even newspapers will write things that aren't strictly scientifically accurate. So always just be careful what the source is, where you're looking at it from. So to get to the questions, what's the difference between the coronavirus and COVID-19? Well, the coronavirus is actually really a family of viruses. They were first discovered in the 1930s, and actually some of the disinfectants that were on sale even before the coronavirus hit the UK actually said on some of the disinfectants, kills coronavirus. So it has been out there. Family of viruses. In December 2019, there was a new outbreak of a new or a novel form of the virus, and that was, as you know, discovered in China. And the whole family of viruses affect the respiratory system. This one was a new strain of the virus, and it's a different disease, one that spreads very quickly. And so the actual disease is named COVID-19. So COVID, coronavirus, it's discovered in 2019, so that's where the name comes. So coronavirus, family of viruses, but COVID-19 is the name of the disease caused by this actual virus. So what's the swab test? So swab test is a test they've been doing since the outbreak of the coronavirus, and that's where they get a swab, which is a bit like a cotton bud, and they put it into the very back of your mouth or into your nasal cavity. What are they looking for? Well, that's where cells will be infected with the virus, as well as in your lungs and so on. And so what they're trying to do, they're trying to remove material from there and send it away to a lab. Now it takes several days, but they will test it. But essentially they will copy the genetic material. The coronavirus is a retrovirus that contains RNA, so they will copy the genetic material, and then they will use gene probes trying to look for the virus's genetic material. So they're actually looking for the viral genetic material from the coronavirus. So what about the antibody test? Well, think back to GCSE, and you know that whenever you get infected, there's various different responses in your body. But one of the specific responses is that the lymphocytes will make antibodies against that disease. So if you get infected with salmonella, your body will make antibodies that are just the right shape to fit onto the salmonella antigens. So we say they're complementary to the salmonella antigens. They're specific. Antibodies are specific, so you're not just looking for any antibodies, you're looking for antibodies that have been made in response to the coronavirus. So the answer to question 3, does it matter what type of antibodies are found? Yes, they're looking for antibodies that are specific to the coronavirus. Question 4, what cells make antibodies? Well, in GCSE you would have learned that lymphocytes make antibodies. If you did a little bit of research, you'll know that actually there's 2 types of lymphocytes, B lymphocytes and T lymphocytes. So I was looking for the answer here, B lymphocytes, which some of you got. Actually, you'll learn that the B lymphocytes, when they come in contact with the antigen, they start to divide, they clone, and they form lots and lots of cells that are called plasma cells, and it's actually the plasma cells that make the antibodies. So the B lymphocytes combine with the antigen, they get sensitized, they divide, and they make plasma cells, and the plasma cells make the antibodies. Question 5, I think everybody knew that, yes, you find antibodies in your blood. How long do antibodies usually survive in a person? It's usually weeks or maybe a few months, that's all. So if you've been infected with a disease and your body has responded by making antibodies, excuse me, yeah, you'll still have the antibodies maybe 2-3 months after that infection, but that's it. It's not the antibodies that give you immunity, it's actually the memory cells, which we'll mention later. So the antibody test is testing for the presence of antibodies. If you've got antibodies, it means that your body has at some point been infected with the coronavirus. If someone tests positive for this new antibody test, what can you conclude? Well, I think I've just answered that. It means that they have been infected, they've had the coronavirus. They mightn't have shown any symptoms of the disease. We say that they've been asymptomatic. They haven't shown any high temperature or cough or anything like that, but yet they've been infected with the virus and their immune system has made the antibodies. Question 8, so if someone tests positive for that, does that mean they're immune? No, we don't know enough. We haven't tested that. We don't know what level of antibodies anybody needs or how long this immunity lasts. So all this test tells us is that the person has had or has been infected with the coronavirus. They may be immune, but we don't know. So it's useful in a way, but it doesn't mean that that person is immune and is not going to catch the disease and they can go back to work and, meet up with people and, go into people's houses and so on. Question 9 then asked you to compare the swab test and the antibody test. So actually in the article, which I have sitting somewhere here, in the article there was a nice little graphic there, and the swab test you might have seen on the news, people driving up and a nurse standing there and sticking something in their mouth. Yeah, so swab test, basically they get this kind of cotton bud put in their mouth or up their nasal cavity. Person needs to be trained to do it, obviously. They have to touch the right place. So it's a relatively easy and painless procedure. So an advantage of that is, it's an easy procedure. Disadvantage is, though, that it takes at least a day to get results. Another disadvantage is that there have been a lot of false negatives. Now, what's a false negative? A negative means no. So, for example, my friend's cousin ended up in A&E and then ended up in intensive care, suffering from high temperature, sore throat and so on. And the doctor was pretty sure that he had COVID-19, so they did a swab test, sent it off to the lab on the Monday. On Tuesday, news came back, nope, it's negative. In other words, you don't have it. But he definitely was getting iller and they were, the symptoms were in line with the ones for COVID-19. So he took another test. It came back on Wednesday, negative. he still wasn't getting any better and still showing the symptoms. So they did a third test, and on the third test, on the Thursday morning, they got the results and it said, yes, so this person has got the virus in their body. So his first 2 tests were negative, but they were false. So yeah, the swab test isn't that accurate. There's some people get false negative results. But it is easy to do. Whereas the antibody test, yeah, you need a blood sample. So that's a bit trickier. Blood sample, but yet you can have the results within half an hour, 20 minutes or so. Basically, the person takes the blood sample and there's a little kit and they mix the things, and you just have to wait about 20 minutes. So an advantage of that antibody test is that it is much more accurate. We're talking 100% accuracy, I think, for positive results. You do get maybe, is it like 0.2% false negatives, which is very small. So it's a much more accurate test than the swab test, but it requires a blood sample. So advantages, I suppose I would say, advantage of the swab test, it's easy to carry out, it's not painful. An advantage of the antibody test, it's really quick. Disadvantage, swab test takes a lot longer and it's not as accurate. You get a lot of false negative results. The antibody test, well, you need a blood sample, and so not everybody likes that, and it's maybe a bit more painful. So, I think I've covered advantages, disadvantages. Question 11 then. Vaccines, what does a vaccine contain? Give 2 answers. So, I think everybody knew that it contained pathogens, but the pathogen obviously had to be either dead or weakened in some way. So essentially you're putting the pathogen into someone's body, the pathogen will have the antigens there and the person's immune system responds to the antigens. But the pathogen has to be harmless, made harmless, so it's either dead or else it's been heated and maybe the DNA's been damaged or something. So that's like a weakened or dead pathogen. That in a mark scheme would only really count as one answer. Some vaccines, however, contain toxins because in some diseases, it's not so much the antigens on the pathogen that makes you sick, it's the toxin that makes you ill, that your immune system responds to. So the other possible answer was toxins, modified toxins. So it would have to be a toxin that has been changed in such a way, still going to bring about immune response, but not going to give you the illness and the fever and so on. Question 12, how does someone's immune system respond to a vaccine? Well, I mentioned there that a vaccine has to have these antigens, and that's what your immune system responds to. So we'll think about antibodies first of all, because that's what we've been talking about. So B lymphocytes are in your blood. There's thousands of different types of them. The one that is complementary to the antigen will sort of lock onto the antigen, and that B lymphocyte becomes sensitized. It undergoes mitosis and it makes a whole, like thousands of cells, most of which are plasma cells, and plasma cells make antibodies. The antibodies then attach onto the antigens, that causes the pathogens to clump together, and phagocytes will digest and so on, destroy them. So that's one way. The other way is T cells. Now that's something that you wouldn't have heard of. And in this assignment, I wasn't expecting you to get anywhere near full marks. It was to get you reading and thinking. So I'm not expecting you to know all this information. And if you got 12, 13, that's very good out of 20. So the T lymphocytes, another type of lymphocyte, again, lots of different types. So one of them will recognize the coronavirus antigen, and that will bring about a chain of reactions which makes the T lymphocyte make like thousands, millions of killer T cells. And from their name, what do these cells do? Well, they kill, their T cells that kill the cells that have been infected by the virus. And they do that by producing a chemical called perforin that actually causes the cell membrane just to have lots and lots of holes and burst. And so that destroys the cell. Now, I mentioned viruses there. Viral diseases and bacterial diseases are very different. If you get a bacterial disease, the bacterium gets into your blood and it multiplies there, and they can actually see the bacteria, well, I don't mean see with their eyes, but they can detect what bacterium is actually there. Viruses are much more trickier to find. Viruses get into your body, and then as soon as they get in, they try to get inside your body cells. And so these viruses are trying to get into the cells in your throat and your lungs and so on. They're only really in your tissue fluid or even in your blood for a short length of time. So whenever they're free and moving about in the blood, which is only really for a short period of time, the B lymphocytes will start to fight against them and make plasma cells and antibodies. Once they're inside your body cells, then it's the T lymphocytes that start to work. And the T lymphocytes, whenever the virus gets into your body cells, it injects its nucleic acid, so the DNA or the RNA gets injected in, but the antigens are left on the surface. So the T cells realize this is your body cell, but it's got this really strange protein, this antigen sticking on the outside, which means it's been infected. So that really gets the T cells working. The T killer cells are made, and they kill your body cells that have become infected. Because once the virus gets into your body cell, a virus will start to fool your nucleus into thinking that its genetic material is yours, and it gets your cell to actually make lots and lots of copies of its DNA and make lots of components, and within hours that cell will burst and release thousands of new viruses. So viruses are very hard to treat and very hard to detect as well. So anyway, I've sort of got a wee bit bogged down with the detail there, but we're talking about B cells making plasma cells that make antibodies. T cells make killer T cells, which then will kill the cells that the virus has infected. One other thing which is really crucial about vaccines and immunity is that both of those types of immunity, the B cells and the T cells, as well as making the cells that help to get rid of the disease, they also make memory cells. T cells don't last in your blood forever. Your antibodies, as you know, only last for months. But the memory cells usually last for the rest of your life in a lot of cases. Certainly they're long term, usually they last for the rest of your life. And that would mean that if you come in contact with the same virus or the same bacterium again, your memory cells will recognize them straight away. And straight away, they will get millions of antibodies made or millions of killer T cells made. And the virus or the bacterium will be destroyed almost immediately when it gets into your body. It doesn't get a chance to reproduce, and so you don't get sick. You don't suffer from the disease. So that is how immunity works. So really in the answer, I was looking for you to talk about the B cells, making the lymphocytes through the plasma cells, mentioning the T lymphocytes and the killer T cells, although I wasn't really expecting you to know that, but you needed to mention the memory cells as well, because that's the immunity bit. Finally, there's a lot of talk about a vaccine, because certainly while the coronavirus is still prevalent, if people start mixing again and it's in the community, it's going to spread unless you're immune to it. Now, if you've had the disease, you may be immune. We don't know. There's not enough research yet. So those people are more likely not to get it. But someone who hasn't got it, somebody who's maybe been shielding, maybe your grandparents haven't been out of the house, if it's in the community, then there is a chance that it will start to spread again, obviously. And so the only way to stop that happening is to develop a vaccine. I asked how long does it take for a vaccine to be developed, and if you Google that, you'll get all sorts of answers, because there's a lot of talk about developing a vaccine against the coronavirus. And a lot of reporters saying, look, we're hoping that we might have something ready before Christmas, and maybe it might be ready for mass production and maybe a year's time. Yeah, but normally it takes maybe 10 years, even 15 years to develop a vaccine. Normally it takes a long time, and so many vaccines get started and they have to go through the lab test. They test it on set human tissue and do lots of testing there before they actually test it on animals, and then finally test it on human volunteers. So there's a lot of research has to go into that before it goes near even a volunteer, never mind used in the community. So quite often it's 10 to 15 years. I'm sure you've heard of Ebola. Ebola vaccine, they started developing it in 2014 and it was only released for use in 2019. So it took that 5 years, which was shorter than the average, but obviously it's a lethal disease and it was spreading very quickly. So they did a lot of work on that very quickly. So they managed it in 5 years. They are talking that a vaccine for COVID-19 might be ready in 12 to 18 months. That's only, that's maybe a very optimistic guess. The reason they're saying that length of time rather than 5 years or longer is because this is a pandemic. Pandemic means that it's a disease that has spread worldwide. These other things like Ebola and SARS and so on, yeah, they hit the news, bird flu, swine flu, and there were hundreds of thousands of people who died, but it was localized in certain areas or certain continents. COVID-19 is worldwide. And so they reckon that with a lot of global collaboration, in other words, the best labs from the best scientists around the world working on this will speed things up a lot. And that's why they're being pretty optimistic about it. So that's what we need to hope for anyway. So hopefully that makes sense to you. I should have said at the beginning, I'm not expecting you to have been taking notes or anything like that. It's simply, to try and help you understand what you've been reading, trying to get you thinking about things. Even when you're listening to the news, do listen to it as a scientist, as a biologist, listen to what they say. I mean, there's been a lot of talk about the R number, and Boris is saying be alert and control the virus. Boris said an awful lot in the last few days, and whilst I'm not a supporter or against him, he has said an awful lot of rubbish as well, because you cannot control a virus. Controlling somebody or something is like controlling a lawnmower or controlling your baby brother, and that's tricky, but you cannot tell a virus what to do. So you can't control a virus. It's not like that. The R number is talking about how quickly it's spreading from person to person and so on. So in other words, I'm sure you've heard good explanations that the R number, if it's 1, that means one person will on average give it to one other person. If the number goes above 1, even if it's like 1.1, it means that if 100 people have it, then in a couple of weeks there'll be another 110 people having it, and so obviously it's going to go up and up in the population. And the fact that the R number has come down doesn't mean the virus has changed. It has come down because people stayed in their homes and didn't go out, or at least the most of them did anyway. And so because we stayed at home and didn't come into contact with anybody, we were keeping ourselves away from the virus. The virus hasn't changed. It's still the same as far as we know. So the R number, it's not that the virus is weaker. It's simply that our social isolating has stopped it from being able to spread. So there's a lot of stuff talked in the news and by journalists and in newspapers and social media that makes a lot of sense, but not all of it is what it seems as well. So just giving you a heads up there. But hope this hasn't been too boring. Feel free to send me videos of yourself if you want to. This has been the first one I've done. So it's downhill from now onwards. So look, I will obviously set you more work. I'm getting you looking at the kidney at the moment because that is usually the first thing we do in September. And it's just tricky. You don't have textbooks because the upper sixth have them. I don't want to be giving you an awful lot of paperwork to print out because I know not everybody has a printer and ink, and it's expensive and it's a lot of hassle. But I'll find some things that will be meaningful for you to do, and I'll probably put a few wee videos or PowerPoints together to get you learning the theory of the kidney and so on. But we're doing well. I'm pretty happy with our progress. But feel free to drop me messages or put something on the stream to let us know what you're up to. And well, you never know when we'll see each other, but hopefully things will be back to some sort of normality in September. It won't be exactly the same as it has been. A lot of people in school have been looking at how we're going to make school safe come August, September time. But you don't need to worry about that. Right, I'll see you again. All the best. Bye bye.